Dr. Nutana, I think once participants are 100 in number, we'll start so that they
Yes,
will.
ma'am.
Yeah.
Shall I start, ma'am? It's 102 participants.
Yes, Dr. Nutana. You can.
Yes. A very good evening to one and all present here. I once again cordially welcome you all to the second session of today's webinar. It's my privilege to introduce the resource person of this session, Professor S.S. Shivakumar. Currently, he is working as an associate professor, microbial genomics at Institute of Bioinformatics and Applied Biotechnology, IBAB. He has carried out his postdoctoral research in universities such as University of Kansas, USA, and Los Alamos National Laboratory, New Mexico, USA. He has also worked as an instructor and lecturer at University of Agricultural Sciences, Bengaluru for 3 years. His current research emphasis is on the application of comparative genomics for understanding horizontal gene transfer and the evolution of bacteria of diverse genera. His area of research is genome-based taxonomy of bacteria. He has published many research papers in reputed journals and has patents to his credit. Now, I request Professor S.S. Shivakumar sir to kindly present his talk on understanding the basic experimental design concepts. Over to you, sir.
Okay, thank you very much, Dr. Nutana. Am I audible to all?
Yes,
Yes,
sir.
Yeah, I think this light is in the background. Let me turn it off. Is this better now?
yes.
Yes, sir.
Okay, thank you one more time for the kind invitation to present in this workshop series, and also thank you again for the kind introduction. I'm very encouraged to see that there are 136 participants, and I hope you will ask me questions during this talk. Feel free to stop at any time. I would like this to be a sort of informal talk in the sense that it's not highly structured, and I intentionally didn't want it to be structured. I wanted to keep it informal because I know there's many of you are students at the master's level, and some could be PhD and others could be at the faculty level. So I also haven't come here as an expert in anything. I'm a learner, and I also would like to learn something from this workshop or presenting in this workshop. So let's get started. I'm going to start sharing my presentation. I have a few slides to show you. So I'm going to share that. Okay. So I have uploaded my slides. Can you see them? Okay, so are you able to see the slides? Can someone say yes or no?
No, sir.
No, sir.
No. Okay, 1 minute. Try to do that one more time.
It is saying that you are started screen sharing, but we are not able to see, sir.
Wonder what happened. Just 1 minute. Let me just get out of Zoom and then get back again. Okay, that could be the problem. So just 1 minute. I'm going to. Okay, I've rejoined. Nuta madam, can you make me the host again?
Yes, I have made. So you're already co-host.
Okay, try that again now.
We can see now, sir.
You can see it now?
Yes.
Yes, sir.
Okay. All right, so sorry for the interruption or delay because of the old laptop. That's the problem. Okay, so I'm going to start here with research design. That's the topic that is assigned. And so madam has tasked me to do this like a workshop talk and not like a seminar or lecture, at least not like a seminar. So lectures and workshop talks usually involve questions and discussions. So I would like to keep it that way. Okay. So getting straight into our profession, our main question today, we have to ask what is research. We all do research, but we have to ask this question. And many times we encounter this question when you say to people outside that you are a researcher or you're an academic, they ask what is research? What does that mean? So there are several definitions, several flavors to our profession, several ways of describing our work. I have put here a perspective that captures the essence of research in a very general manner. So research, as most of you know, is a very generic term. It covers all kinds of activities or studies that are designed to find some outcome, some response, or some effect to questions that we are interested in. And we would like to obtain the responses or answers or outcomes through ideally systematic and scientific approaches. So what is the difference, systematic and scientific? Not all scientific approaches are systematic. They need not be systematic. And not all systematic approaches. Say that again, sir.
Sir, slide ಕಾಣ್ತಾ ಇಲ್ಲ.
ಕಾಣ್ತಾ ಇಲ್ಲ.
Yes, sir.
First slide ಇದು ಕಾಣ್ತಾ ಇದೆಯಾ ಸರ್, ರಿಸರ್ಚ್ ಡಿಸೈನರ್?
ಇಲ್ಲ ಸರ್, ಕಾಣ್ತಾ ಇಲ್ಲ ಸರ್.
1 minute. ಸರ್ ಈಗ
ಸ್ಟಾರ್ಟೆಡ್ ಸ್ಕ್ರೀನ್ ಶೇರಿಂಗ್ ಅಂತ ಕಾಣಿಸ್ತಾ ಇದೆ ಸರ್.
ಮುಂಚೆ ಬಂದಿತ್ತಲ್ಲ, ಇಟ್ ವಾಸ್ ಶೋಯಿಂಗ್ ಫಸ್ಟ್ ಸ್ಲೈಡ್.
ಎಸ್ ಸರ್, ಬಂದಿತ್ತು ಅವಾಗ.
Wait.
ಈಗ ಕಾಣ್ತಾ ಇದೆ ಸರ್.
Okay, thank you. So I will not put it on slideshow. One problem on the slideshow, no problem. So I'll just keep it like this. I hope
Okay,
it
sir.
is okay.
Yes, sir.
Okay, so let's get to that question. So we want to obtain answers through scientific and systematic, ideally both, okay, approaches. So in the realm of the research, clinical trials also are important because they provide significant data or information about new drugs, new vaccines, new medications. So clinical trials are also part of research. And clinical trials, as you know, involve human subjects. So they have to be even more systematic or scientific than other types of research that we do in the laboratory. And what about surgical procedures? These are actually procedures that involve experimenting upon a human subject. For example, there may be a new surgical procedure that has been developed. For example, in cardiac surgery, you have to do it on live patients. Are they part of research? They are also part of research, and they are more riskier. They are more costlier, and there cannot be any room for errors there because they can lead to huge problems. So I have put them in the order, like the first paragraph there and answering the question, what is research? That is like the mundane or most of the work that we do. You know, we want to find a response. We want to do an experiment. We're working in the lab. Of course, we want to be systematic and scientific. But the next level is clinical trials, very important. We have to be careful with clinical trials. So even the next level is surgical procedures, improving a surgical procedure or deploying a new surgical procedure into the hospitals or operation theaters. That requires even more rigorous systematic and scientific approaches, okay? So that is how research is designed or defined. So we want to obtain meaningful answers to some questions or some curiosities that we have about nature or about some molecule or some procedure or some application. Then the key question is, why do we do research? Anybody would like to just take the bite and answer this question? Why do we do research?
To know something new about already existing thing or theory or concept.
Correct. So fundamentally, what drives most of us to the research field is curiosity. Curiosity about the natural world, curiosity about the living world. In our cases, curiosity about, animals or plants or insects or, microbes. So we are driven by a passion. We are driven by inherent curiosity to understand nature, to understand how things work. Okay, just so research is an all-encompassing subject. Astronomers do research. They want to know what exactly happens in the outer space, the astronomical realm. How do stars align? Why do we have seasons? Why do we have climate change? And that is far-fetched. I mean, that is too far. That is beyond our planet. Most of the research that we do, at least as biologists, is plain and straight, simply on this planet. We cannot do, most of the cases, we cannot do anything outside this planet. There is exoplanetary biology. There is sequencing done now in the space station or outside of this planet, but that is very minimal compared to what is done here. So we are very grounded people, very down-to-earth people. So we're interested on phenomena that happen on this planet. So why do we do research? Because we are curious. We are driven by a passion. But when you take up research, if you are working in universities or colleges, part of your work also is doing research in addition to teaching, which means it is, let's be honest, it also, provides you income. It's job security. It's also source of income because you're doing research, your university is supporting you. So I am honest with that. It's not that many people do research because they will get paid or they are employed or they get a salary. It's actually secondary. They start out as being teachers, they become researchers, and the universities or colleges like their work, so they get a remuneration. Of course, that is part of doing work. Nothing comes free. But in the old days, if you looked at how research was done, it was done without any remuneration. People were simply curious. They found the tools and they did work and they discovered many good things. And I don't think they were compensated. Most didn't even get a recognition. But these days, research comes with enormous responsibility, recognition, and of course rewards. It's all part of it. So why do we do research? Basically, this is how we can answer that question. First and foremost, we want to gather or gain some evidence, some support for some of the ideas or theories that we have in mind, or we want to provide some evidence for some practice that needs to go into the field. For example, as I mentioned before, some surgical procedures, something that has to be done, performed on animals or humans. So we need to have some solid ground before we can deploy some procedures or some medications or vaccines. B, we want to inform people who are into the action, which means our research, our work should be able to allow people who are in the decision-making chairs so that they can make what is called informed decision. For example, when the Omicron strain of coronavirus came into the picture, we had people at both the political and bureaucratic level sitting and talking, but they were looking for answers from people who actually had worked with the coronavirus, so they can provide some input. So that input is based on some research. Some research is going to tell people, the decision makers, about how do we avoid, how do we minimize the risk of the virus. C, contribute to developing knowledge in a field of study. In research, we stand on shoulders of others. There is no what we call vacuum research. We don't do research in a vacuum. We cannot do research in a vacuum. We cannot do research in isolation. It's unthinkable. It's preposterous to think that someone can do any research without being in contact with others, without networking with others, or without support from others. This is a field that heavily borrows from others' work, and it contributes little, very little. But that little is what adds up. If everybody contributes a little, it's like a drop in the ocean is nothing, but every drop there counts. So everybody adds a drop to the ocean of knowledge that we have. So those are the three main reasons why we do research. But that is when considering research as a profession. But our life itself can be a research, because if you know, many people don't realize, but everyday life is also research. Whether you cook in the kitchen, you repair something at home, or you're simply watching a documentary, you have to do some research. What kind of documentary should I watch? How did they do this documentary? Is it worth watching? All of that involves doing some thinking, some analytics. That is part of research. You may not realize that. So learning to conduct research is an integral part of learning about life. And I extend that to saying learning to conduct research is an integral part of learning how to live a life, a meaningful life. You may not be a researcher in the defined sense of research as an academic or a university professor. Even a common man, even farmers, when they sow seeds, they would have knowledge accumulated from years of work, and they may not have documented it, but they have memory that tells them when exactly to sow the seed, when to harvest, and when to market. Of course, they failed miserably in the last one, but at least they have some knowledge which comes from research. And that knowledge was added or contributed by all the people in the community, bit by bit. As I said, every drop counts in this field, okay? So I borrowed these 7 points about why we actually do research, although I tried to put those 3 or 4 things before. This is very new. I was reading an article, and again, that was, I had to update myself, and I found this article that was published last year, 1 year ago, in February 2021. And I just borrowed some material from that article. So 7 reasons why research is important, whether you are a common man or you are an academic. Number 1, research is what helps us build the knowledge base, and also it facilitates learning for ourselves, and also research is what we use as a basis to do teaching. There is no textbook that can be written without the research base. So all knowledge is research based, whether it's textbook or journals. Number 2, research is a means to understand issues and increase public awareness. Of course, as I gave the example, the coronavirus situation right now requires our understanding about the virus, its spread, and also to increase public awareness about how it spreads so that we can mitigate the risks. Number three, it helps us succeed in our profession. That is very selfish, but we succeed in our profession because we are into asking questions that are going to provide some answers to make society better. Number four, it allows us to disprove lies and support truths. Most important, you may have watched several science shows which actually want to dispel what is held as a belief or superstition. Those are very much important. Unless we have some research basis to dispel that, people are going to hold on to that truth very strongly. So in many cases, we want to dispel that. Number five, research is a means to find, gauge, and seize opportunities in the natural world. So we discover something that is going to provide us some product, some gain, which can be used for society. So the best example is the Green Revolution. So there are better crops that better seeds were developed. So that is an opportunity that the world seized all over. Otherwise, there would be so much famine, there will be so much hunger without that research into having better crops. Okay. So research promotes love of and confidence in reading, writing, analyzing, and sharing valuable information. So the sixth one is what is most appealing to me, because this is what we do on a daily basis if you work in a college or a university. It provides us a love for reading, a confidence in reading about something that is published. It also pushes us into writing. You have to write. You simply cannot read and sit. You have to write about it. You have to at least make slides, as I did, to explain it to other people. Of course, being analytical is part of doing research. And most important is sharing valuable information to disseminate the knowledge, disseminate what we have learned through our research, whether it is experiment-based or simply observation-based. Last one, research provides nourishment and exercise for the mind. Most important. A lot of people have no retirement in research field. They continue to work into their early 70s or even up to the late 70s or even early 80s, because they develop this acumen or there's the skill for doing research, and they're mentally very active. So it's like teasing your mind every day, because that is nourishment or nutrition for the mind, or at the end of the day, it's nourishment for the brain. So you want to keep yourself active both physically and mentally. So reading, writing, and being analytical is part of that active life. Okay. So now I come to the main question of the topic that is assigned. Why should anybody care about research and research design, especially? Whether you are a researcher or whether you're someone who don't do research but would like to disseminate the knowledge. Let's say you are a press reporter or a journalist. You're trying to disseminate the knowledge that has come out of research. You haven't done that research, but you are trying to disseminate that, or you're simply doing research, or you're a student who is still learning. So why should anybody care about research design? Again, this is an open question. Anybody?
Because it is a blueprint of research.
It's a blueprint for research. Correct. That's a blueprint for research. Any other answers? Anybody else? I want one more answer. Yes, I agree with that. It's a blueprint for us. It's actually a document, or it's a commitment, actually. Anybody else? Why should anybody care about research design, especially us trying to do research? Anybody? There is more than 120 participants. So 1 out of 120 is less than 1%. Let's make it 1%.
We can do a very systematic research.
Say one more time, madam.
We can do a research in a very systematic way, sir.
Correct. If you have a better design, you can do a systematic work. It's not about just research. It's, everyday life. If you have a better design, let's say you have a better vehicle that is well designed, then you can drive it better. If you have a road which is well designed, then you can drive better. So same is about research. If you have a well-designed research, then you're going to do better work with that. So the answer to that question is research design, which I have put in bracket scientific method, because they're inseparable for us. Scientific and systematic work is inseparable from research design. So research design is the key to success. So successful driving without accident requires a good vehicle, good driver, and good road. So similarly, research design is the key to success because number one, when you design your research, you want to keep all assumptions out of the equation, out of the boundary. Again, I want to add here, you cannot keep all assumptions out. You have to have some assumptions to do research, okay? We want to keep all unfounded or unsupported assumptions out. You want to keep supported assumptions, meaningful assumptions into the equation. Very important to keep some assumptions. We might call them hypotheses. Number two, the design helps us to bring in good methods or good practices into our work. The best, they may not always be the best practices. So I deliberately avoided the word best here. I didn't put the term best because that's scary. Let's not be perfectionist. Let's not be best all the time. It's good enough to be good. So we want to have good, we want to bring in good methods for doing work. Number three, most important, avoiding biases. We want to avoid research bias. Best way to avoid that is to have good methods, to have good assumptions, and keep the bad things out of the equation. So of course, will we be biased or unbiased all the time? No, it's very difficult. So that is a tightrope walk. If you ask the question, are all researchers unbiased? No. There will be some bias in everything that we do because we are humans. Humans are inherently biased. And if you are unbiased, then you have to be a robot. You have to have machines doing. Of course, we do instruments, we do use robots to do work. But at the end of the day, the research is analyzed by humans. And so you have to put that word so that we have in our conscience that the bias is like a mosquito that is lingering around all the time. You cannot get rid of it. If you get rid of one bias, there will be another bias. If you get rid of one mosquito, there will be a few more mosquitoes coming. So bias is something that we have to be very careful, but we should also know that there will be bias somewhere, and we should know what that bias is and be able to tell that there may be some bias here and there so that people are aware of that. So let's consider some facts or some points about how to go about a research design. Why should anyone care about research design? Number one is, what is the question? What question are we asking? What are we interested in? What is our curiosity about? And also, how big is this curiosity? For example, we could be curious about some planet, some star that is millions of miles away. That's a big question for someone on this planet to answer. But it could be a question as small as a virus that is on this planet, and it's a very small entity, invisible, but the questions about that virus could be very big. So the term here, big, is a relative term. The size of the entity could be small, but the question about that entity could be very big. The size of the entity could be very big, but the questions about that entity could be very small. So the big or small is a very relative question, but we have to be aware how big or how small is the question. Two, are the goals achievable? Are our objectives achievable in the circumstances that we find ourselves? What is the circumstance? We could be working at a small college, or we could be working in a full-fledged research institution. So we have to be very careful about setting our goals. You can't set your goals, or you can't ask questions that are too big that cannot be achieved or answered in the circumstance we find ourselves. Let's say you are in the best equipped research institution, but we may not be able to achieve some goals because circumstances are such that you cannot go to the lab every day like we are in now, because we have to work with the pandemic. So are the goals achievable in the long term? That is what is important. Long term, not in the short term. Number three, what level of information will it provide? After having spent months and years and lakhs or maybe even crores of rupees, if it doesn't provide any useful information, then what was the point of doing this work? Was it worth doing this research? The information that is obtained is, if it is superficial, if it is an information that nobody relies on, nobody trusts, it doesn't build any reputation, then what's the point? So we have to be very careful about the third point, which is the level of information that it provides. We have to keep that in mind before we design it. The key question is, just ignore the first 3 points, just ignore the 3 points next to it. The key question for all of us as humans is, is the research design Is the research that I am going to do suitable to the level where I am at? And what does this mean? What does this last question mean? Is the research I'm going to do, is the question that I'm going to ask, is the research design that I have on paper, is it suitable to the level where I am at? What is the answer to this question? What does it mean to ask this question? The question is the level where I am at. Any answers to that? What do I mean? I know what I mean, but I want to know whether you understand what I mean here. Level where I am at. Ask this question to yourself. Level where I am at. What does it mean?
Either design is fundamental design or experimental design.
Correct.
I think it will be clear.
Right? Anybody else? Level where I am at. This is a very personal perspective about ourselves, about myself, about yourself when designing research. Where am I at? To answer this, I will come to this chart, this graph. This is very neatly illustrated. On the y-axis, we have interest or curiosity. On the x-axis, we have what is called practicality or feasibility. And if you look at the graph or how the curve bends downwards or goes upwards, to the extreme right, so the feasibility increases if the problem is easier and takes less time to address. That's what feasibility means. Whereas the feasibility decreases if the problem is too hard or if it takes too long to answer. It's like a nut to crack. If it is a peanut, you can easily crack it. That's what an easy problem is. If it's a walnut, it's hard to crack. You require a stone to crack a walnut. It takes more time to crack a walnut. So that's the easy or the hard problem is the peanut and the walnut. Okay. And on the x-axis, interest or outcome or curiosity. If it is a small gain in knowledge, then that is what a hard problem is going to give us. It's incremental. The hard problem that we are trying to address may not provide all the answers or solutions in a short period of time. But over a long period of time, it might help us accumulate a large body of knowledge. Okay. And the large gain in knowledge cannot be obtained if the problem is easier. So if you see the stars indicate who should pursue what kind of work. So that is the question that I was asking in the previous slide. Is it suitable for me at the level that I am at? If I am an undergraduate student, a BSc student or a BTech student, I cannot be pursuing a question or problem that takes 5 years to answer, or it requires too much input. I have to pick a problem that I can answer in 1 semester or maybe 1 year. Okay. So if you're a master student, then it's a problem that's slightly less easy. I wouldn't say difficult, it's less easier. If you're a PhD student, the problem is slightly more harder. It keeps on increasing. The hardness increases or the feasibility decreases, but the interest level may increase because you want to pursue it over a long time. So if you put all of this together, then that is what you are, or that is what you would be if you are a university professor or a college professor. So problems that are hard, that take long time to address, that will accumulate a large body of knowledge are good for professors or people who want to spend an entire career trying to address a system or develop a system or a model organism or a model procedure. So the red stars indicate things that are not considered good problems for someone who is going to invest short amount of time or less amount of resources. The blue stars indicate problems that could be addressed by people depending on the resources and time that they are willing to invest in addressing the question or developing the research methods or having a research design in hand. Okay. So I hope that graph was helpful, because if you look at this, you can use this graph to tell students when they come as interns or when they come as master students or PhD students that they have to be aware. They have to have some practical understanding about how to design a research based on the situation or level where they are at. Of course, you're not going to show this to a professor or someone. Of course, early career researchers or those that have joined employment recently would still benefit from this, so that they know that they're going to be addressing some problems over the long term. Okay. So now, what is the roadmap or what is the path to have a good research design? Again, I didn't put the best or the perfect. It's simply what constitutes a good research design. Number one question, of course, fundamental is always, why am I doing this work? It's not because it keeps me employed, or it is gainful employment, or it gives me some salary. It's a fundamental question because we want to address a problem. That is as clear, as simple as that. If we don't want to address a problem, then there is no point in doing research. If you want to address a problem that has already been addressed, then that is duplicity or duplication. We don't want to do that also. So number two, are there unknowns? If there are unknowns, what kind of unknowns they are? Which means if there are unknowns, most likely somebody has already tried to address them previously and just left them because they couldn't address them. And we have to be very careful there, because if they had difficulty in addressing, then we may have to be very careful whether we would be able to address them or not. So we have to pick the unknowns very carefully, which means you are not shooting for the stars or the planets that are beyond our reach. You have to shoot for something that is within our reach. So the unknowns should be clear. At least we should have some understanding of what is unknown here. Number three, based on the unknowns, what kind of aims or objectives can we have? So our aims and objectives can be very broad. Now, once we have the aims and objectives, we may have to have some hypothesis. And we're going to break down the whole problem into several smaller projects or several experiments, which each one may have a hypothesis. So what are the hypotheses that we're going to have? Number five, at the end of all of it, what would be the outcome? What are we going to deliver to our institutions, our universities, our colleges, or to the funding agency, or to the public taxpayer? Number six, what resources are available to address the unknowns or to accomplish the objectives? As I said, working in a university or college is different from working in a research institution, because people there have teaching responsibilities. They may not have all the resources that a full-fledged research institution, which is well-funded, may have. Okay. Number seven, what is the time frame? What is the duration? I'm an undergrad student. I have only 3 years. I'm a PhD student. I have maybe 5 to 6 years. I'm a postdoc who may have only 3 years or 2 years. Or I'm a university professor who may have employment for 20 or 25 years or 30 years. So depending on the time frame or duration that is available, we have to design the research project. So once we have that, can I write all of this and have it on the drawing board, or have it on the wall, a research model that is drawn up that everybody could see? So this is the model that I'm going to work with. Of course, it is not the final. It's always going to be a draft until I finish it, but at least I have a model. Number nine, what are the risks? What are the risks in doing this work? For example, if you're working with animals, there may be risks of disease transmission from the animal to the researcher, or vice versa, zoonotic diseases. Or if you are working with a virus that is highly contagious, the people or person who is going to work with the virus itself might contract it and may succumb. That happened with Ebola virus. Doctors who were treating patients died because it was highly risky to treat them. Okay, so we had to find ways to mitigate them. So how to deal with the contagious viruses? We have PPE, personal protective equipment. That's the easiest way. Or if you're working in a laboratory, wearing a lab coat is the simplest way of mitigating a risk. It will avoid 4 or 5 kinds of common risks. Okay, and wearing gloves, of course, is another very generic or common way of avoiding risk. Number 10, most important, is there a plan B? What does it mean when I say here, is there a plan B? Anybody? What is the context here about the plan B stuff? What do we say when, or why do we say, is there a plan B? Anybody? What is plan B for a research design?
Blueprint of the study. Blueprint
of the study. Yes. Anybody else? Plan B. Plan B, if everything I have designed, if everything I have done fails, what am I going to do? I'm going to just leave, or I'm going to just quit, I'm going to disappear? No. Plan B is, you want to have 2 or 3 parallel projects or research designs. One blueprint is not sufficient these days. One blueprint is simply not enough. One experiment is not enough. One design is not enough. So plan B means that if something goes wrong, if you encounter a huge risk, it doesn't mean that you hit the point zero again or you hit ground zero. No, you drop down to a middle level and then you pick up and move again. It doesn't mean that if everything fails, you go down to the bottom and start again. Plan B helps to avoid you from getting down to the bottom. It keeps you afloat at some level so that you can again soar to the top. Okay, plan B is very important, which means there is a backup plan. B is backup. Okay, so now I come to this question, or let's say research design is all good, but then we should, once we know what is research, we should also know that there are different types of research. Of course, this could be, some of you could be well versed with this. I could be repeating some of this stuff, but based on my own experience, my own work, I look at research in types of research in different ways. So let's look at the different types of work that could be done by different groups of people that I showed you in the chart. Okay, the less feasible versus more feasible, the less curious or less interest versus more interest. So the research could be of different types based on the chart. So types of research based on goals or objectives or what needs to be accomplished. So there could be exploratory research. This is what we call groundbreaking. Nobody had done work like this before. So that is exploratory. For example, discovering that the novel coronavirus has a huge genome. That is exploratory because previously we knew that coronaviruses don't have that kind of a massive genome size. So that is something that someone explored and found out that it has a huge genome. To conclusive research that is being pursued so that you want to conclude something which wasn't concluded better or wasn't concluded at all before. For example, knowing that coronavirus keeps on mutating, to tell the world that this is a virus that will keep on mutating. That's conclusive. We now know that it's not going to stop. It's going to saturate once all mutations are done or all mutations are saturated. But most of the time, our research is not as clear-cut as shown there. It falls in the middle, somewhere between being exploratory or conclusive. So I would like to call it exploratory conclusive because you're on the way to exploration, but you hit a point that this is conclusive. Okay, so you have concluded something. Of course, it may not be the final conclusion, but you are going to have some conclusions that convince people that yes, this is correct. So based on the methods or tools that we're going to use, research could be qualitative, which means you're not going to be very analytical. You're accumulating some data and you're making that data available to others. You're going to also show how that data was gathered. It could be quantitative. You're trying to address some hypothesis based on some population, some groups, control versus experimental. So that is quantitative. But again, that is not as clear-cut as that, at least now in the genomics era. A lot of our research starts being qualitative, then it becomes quantitative because we would have generated some hypothesis based on the availability of some genomic data, but then we would accumulate a lot more data by sequencing more genomes, so it becomes quantitative, so we will address some questions that way. So again, it merges. And types of research based on the outcome, most important for us. What is the outcome of, as I said, having done all of this, is there an outcome that is useful, that is meaningful, that will convince people to fund it further? So if you are working in an industry or a private company, most of the time your goal or your outcome is whether the research is commercializable or something that can be productized, that you can make a product and sell. So you have commercial interest, you have business interest there. You may not be interested in publishing it. You may not want to reveal it to the world. But if you work in a university or work in public funded organizations, they're looking for publications to show the world that the work has been done, and it may be commercialized or it may not be, but at least it has been published, that the taxpayer knows, that the other people know that the work has been done. But again, even in universities or research institutions, we are looking for now to make products. So our research is first publishable and maybe eventually commercializable or productized. So it merges somewhere there. So that is how I look at different types of research. Okay. Any questions so far? Anybody has anything to ask? I want to hit the pause button at this stage because
excuse me, sir.
Yes.
Commercializable, we can say this generalizable. We can generalize it
In what way? If you
Generalize whatever outcome, we can generalize the research result. Can we say the commercializable?
So what I mean by commercializable is that our research has yielded a product that we are now going to sell and make money. That's what I mean.
Okay.
But you may not be interested in publishing it because you don't want competitors to know. Okay. But in the academic arena, we are not into competition. We are into collaboration. So we want to publish and we borrow stuff and we give stuff to others. So we are not actively competing in the research arena in universities or colleges. Okay. All right. Now I come to the most important part of the talk, which is there a difference between research design and research method? Because somebody was trying to say something. Can we have that one more time, please? No?
Yes, there is a basic difference.
Yes, please.
The research design is what actually we are going to do our research on, based on, and the method is what I'm going to do stepwise according to the research design.
Exactly. Very, very correct answer. Okay.
What and how? We can say it what and how. Research
Correct.
design is a what, and research method is how.
Oh, exactly. Very good. So this question, and the answer to this question, I took it from the university where I did my PhD work in the US, Virginia Tech. So I was looking for this, and I was going to the university website to find something. So it was on the library website of the university because they actually conduct courses for the students as part of the library exercises. And I had actually taken a course during the first year of my PhD. So, and they have actually improved upon the original course, which actually used to be simply research methodology, but now they actually make the course as research design and research method. So answering this question, what is the difference between research design and research method? Research design is a plan to answer your research question or answer your curiosity. So as somebody said, the research design is the what. But the research method is actually a blueprint about how we're going to do the work or how we're going to implement our plan, implement our design. Okay. So design is what comes first. Design is much more broader. It's like broad brush strokes, but the method is fine tuning. So there may be 5 different experiments or methods available to answer a question. Which method are we going to ultimately choose to answer that question? Again, which method we choose depends on our context, the resources available to us. The question could be the same, but is addressed by 2 different groups or 2 different people located in 2 different institutions, but they might come up with similar answers because the question was the same, their approach was the same. They simply use different methods. Okay, of course, the outcomes need not be the same or similar all the time, but methods are very carefully chosen. They have to be fine-tuned to obtain the correct answer. If you are trying to obtain an answer about what proteins are expressed in a model organism, you have to choose the proper proteomics tool for that. You are not going to choose a tool that is going to be transcriptomics. Why? Because transcriptome will only tell that the gene was transcribed. It doesn't tell you that the protein was actually made. That's where I see the problem with DNA microarray. DNA microarray is a very powerful tool or method. It will tell about the activity of the genome. It tells how much percentage of the genome was actually transcribed. But the problem with microarray is it doesn't tell whether those transcripts were converted into proteins. That's where proteomics come into picture. Of course, would it help to have the DNA microarray data transcriptome? Of course, yes, it does help. But if your question is about what percentage of the protein genes are converted into proteins, you want to fine tune it to the level of proteomics. Okay, that is what it means to have the method or strategy to implement the plan. But again, it's a tightrope walk. You cannot draw a line or be very strict about the design and method in the context of research. Why? Because they are, of course, different, but they are so closely related that when you are designing a research, you are, of course, thinking about the methods or tools you're going to use. And the methods and tools that we're going to bring in almost always depend on our research design. So they go hand in hand. It's like having two hands and working instead of having just one hand. So good research design ensures that we obtain good data, and to obtain good data, we need to have good methods. And to bring in good methods, we have to have a good research design. So they're inseparable in that context. Okay. So now, what would be some of the most frequently used methods? I already gave an example in biological sciences, at least in the last 10 to 15 years, or even maybe 20 years. The tools that we use have become more molecular biology based or more data based. So we work with genomes, we work with transcripts, we work with proteins, and we use all kinds of tools in biological sciences these days. But if you ask this question to an undergraduate student, let's say they have just come out of 12, they are about to finish their undergraduate education, ask this question, what would be the most frequently used research methods? Let's see if we can get some answers to this. What are the methods that anybody has used? Let's say many people here are researchers. So I want to hear from you. What method or what tool have you used? Some volunteers, please. No answers? Of course, you have done research. You have used some method. I already gave an example. DNA microarray is a method or a research tool. So like that, I want some answers. Very generic, undergraduate level.
The interviews and surveys.
Okay.
Louder,
experiments. Louder, please. Louder.
Experiments, interviews, surveys.
Yes.
Questionnaires.
Yes.
Questionnaires.
Yes. Anything else?
Structured teaching method, sir.
Say that again.
Structured teaching methods.
Okay,
STP.
correct. Yes, STP, yes. In teaching research, yes. In academic pedagogy research, yes. Okay, so all of those are correct. The most fundamental type of research, if you ask 200 or 300 years ago, how did our ancestors, and I say ancestors, these are scientific ancestors, not our biological ancestors. So how did our ancestors do research? How did they work? Of course, most of that was observation based. Darwin's work was observation, collecting specimens, of course, a little bit analytics, but a lot of that was observation and critical thinking. So observation is the key to all work. 2, of course, some of you mentioned surveys, survey-based research. 3, interview-based research. Surveys don't involve interface between humans, but interviews do involve human interface. Focus groups are one type of research. But for us in biological sciences, methods are experiments, experiments are methods, or experimental methods are key to our work. So I put that in red so that we are all aware that our work could be based on observation, it could be qualitative, but it will get quantitative at some stage or the other. So we want experiments. We want a design that will involve experiments. Okay. So number 6, secondary data analysis. Again, more common in our field because we have data that has come from different groups, different studies, and we want to combine that data and see if all the data agrees, or if one set of data agrees with the other. So that is secondary data analysis. So that is conclusive research that falls into the realm of conclusive research. So we have a lot of data-based research in the genomics era. Archival study, for example, a lot of data was collected. A lot of data was collected a long time ago, but nobody worked on it. They were so busy collecting the data, they just put the data somewhere and then they forgot it, or they didn't have resources to do the work. So we want to go back and dig up and do some archival study. That is, and it also involves going back to people who were buried long time ago to explore what kinds of diseases that they suffered from. Some people went and found that people who died 2000 years ago had Yersinia pestis, a bacteria that causes plague. The Yersinia pestis was found in people who had died 2000 years ago. Same thing with tuberculosis. They could find the genomes or DNA of Mycobacterium tuberculosis from well-preserved mummies. Okay, so that is archival study. Okay, going back to what is available. But what is most important is to know that methods are now mix and match. you cannot simply have one method and do any meaningful work. You have to have several methods. Of course, as I said before, it starts with observation. Then you're going to have hypothesis. So it's a mix and match. You're going to have animal experiments, you're going to have laboratory experiments, you're going to have sequencing data, you're going to borrow from data, you're going to borrow data from other people's work. So methods, if you read a typical research paper now, it's a mixed bag of methods. They would have used different kinds of methods. Research has become so complex now that you cannot address a question meaningfully unless you deploy several methods. If you see some papers, there is at least 20 different methods. The methods section is so complex that they have to reference several previous pieces of work to help people understand what method they have used. Okay. So just know that we as biological science researchers use so many methods. Simplest method could be having polymerase chain reaction or PCR or sequencing. Sequencing is one method that we frequently use. Okay. Now, I've come to the end of the talk, but how much more time I have, madam? I started around 5:35.
So this session is till 7 o'clock, but you can take up to whatever you time is required.
Okay, so I have only 2 more slides. I also have a short exercise for the group participants, but I want to show you a short video. This is from an Indian researcher. So I deliberately avoided bringing perspectives or context from people outside of India or outside of our own working conditions, because I find them too distant for our environment or our context. So I attended a workshop come training session for early researchers, early career researchers in 2016. There I met a researcher called Ramnath Bamezai. Some of you may have heard of him. He's a cytogeneticist, molecular biologist, cancer biologist. So I liked his talk, and I had some words that he spoke that they have stuck in my mind. So I'm going to share a brief. So the entire talk is about 45 minutes. We are not going to watch the whole talk. I will show you parts of his talk that actually relate with what I spoke today. Okay. So do I have permission to share that video, madam?
Yes, sir, you can.
Okay. So I will only show the first 10 minutes of the talk and then the last 2 minutes. Okay. Okay, so I have shared the screen. Is it visible on your computers?
Yes, sir.
Okay, fair
It's
enough.
visible.
So the title of the talk was competition with its highs and lows and a struggle to make a difference in the biological sciences. This was a talk given at the 8th Young Investigators meeting in 2016. It's still relevant. You can watch it every day. It just inspires you. So let's see if the audio plays on your computers. If the audio doesn't play on your computer, just let me know, okay?
It's been a pleasure.
Does the audio play on your computers?
Yes, sir.
Okay, excellent. Okay.
3 days over here. In fact, I realized after thanking the organizers and Dr. Dinkar, who sent me the invitation to begin with, and then obviously India Bioscience team and others who are associated with this. It has brought me to a learning curve and added to that learning curve. And that brings me to the first lesson, which all of us have in our mind very predominant, and that is the learning curve growth does not stop anywhere, it doesn't saturate. And if it saturates, that means that we're not contributing to science. So it has definitely been very fruitful time over here these 3 days, and I've learned a lot. Now, besides owning the job of mentorship, I thought probably I was a mentor also. I was getting mentored during this process. I belong to a category which is a little different from the categories of scientists whom you heard as mentors, because I'm homegrown. And obviously, when you start a career, it has lot of difficulties. It has a lot of stress associated with it because of variety of reasons, which will come in a minute. So I belong to that category of, I'm not a prodigy, and nor do I have a very great pedigree. Because in Indian science, when I entered in early 70s, the pedigree requirement was it still continues to be there, but it was very important. Who's your mentor? And which lab you came from? And what's the name associated? So I just put it funny over here. Famous boss to hold your hand, to promote your future. India, somehow we used to be in that kind of mental state. So what was important for me, if I started my career, research career in India, and I continue to be here, though I went abroad several times afterwards, establishing the credibility became a priority for me. So lesson number 2. First was that the curve, which is learning curve, it does never ends. It does not saturate. And second lesson is that it's very important in science to establish the credibility. And you all know what all goes in establishing the credibility and continue with that. And I did not run away, but I ran against the current and against the dominant voices which prevailed and prevail till date. Because you have dominant voices which run the show. It's not most of the times the dominant voices may not be the right voices. So what is important to learn here is how to contribute to that dominant voice so that along with it, there is another voice also emerging and then it's heard to some extent. That's what changes the policies. That's what changes the basic character. So challenge was at another level, the challenge was working in the university system. Today, university system is becoming much better with great funding. Earlier in my days, 4 decades back, the university system did not have infrastructure, it did not have adequate money to support research. So 4 decades back, it was resource and infrastructure, and then you have responsibility of teaching, and your vice chancellor puts on your shoulders other added responsibilities of some administration here or there. If you don't get house in the university, then you are made a warden of a hostel, which is another problem to deal with the students. So you have added responsibilities in the university setup. Nevertheless, I think it is normalized over these 4 decades. It has normalized now. We have a number of 16 new universities have been added, which are central universities, which have the same status as JNU, Delhi University, and other central universities. So you have more opportunities to enter these universities. And I'm sure I just came a day before I came over here. I had gone for some selections at Central University of Kerala, a wonderful place to visit. And we inducted some professors, associate professors, and assistant professors. And I saw one young investigator who had joined last year this program in Kashmir. So he was there. And then when I asked him, who motivated you to come for this interview? And he gave me a name and we interviewed him. So I have chosen this topic, although I'm going to wrap it along with my research activity and convey that what difficulties one faced, what were those highs and lows and the struggle to make a difference in one's own way, because ultimately it's your own conscience which has to be satisfied. You have your peer group to review you, assess you, but ultimately it comes down to, if you're an honest person, then it comes down to your own conscience. Are you doing right thing? Are you ethical? So ultimately, I believe in that Gandhian paradigm. Maybe I'm old enough to believe in that kind of paradigm, but then it's to make a difference, it's ultimately your own conscience and continue with that. But over the years, research became a passion. Today, I think that 65 years of my age, I should just wrap it up in a capsule and give it to a youngster in my area of research so that he just grapples with it and within a day or two, knows about everything and starts from there. The career starts from there. Now imagine this 25-year-old or 22-year-old youngster starts from all that what is known till 2016. The questions are going to be very different. The questions are not going to be those repetitive questions which I addressed and grappled with and moved on. I think mentorship is that. For me, mentorship is becoming a challenge for me for future years because I have refused to continue in JNU, Jawaharlal Nehru University, because I thought, let me contribute in some different way. And since I have trained 400 trainees in the past 10 years, it's passion with me to train people and to expose them whatever knowledge base I would be equipped with. So it has become a passion, but late in my career because I had to establish credibility first. I don't know whether I have established that or not. I don't know, but it has become a passion. And what's important is, since I did not have great mentors, the time period was like that in India around that time. So how did I learn? I learned by carefully watching people, listening to people. Now this is another lesson. For you all who have done postdoc from Yale to any other institution, Harvard or others, they may not think it's very important, but I feel it's important for them too. Observing, listening carefully, learning, because learning is there from everyone. And then you can filter at your level that what you want to imbibe or you don't want to, you want to throw it away. And the world outside was obviously a feeder for me, mentoring me, and keenness vision, which mentored me over the years. Unfortunately, I did not have mentors. This is my career graph. So I worked on a problem, PhD problem, prenatal diagnosis. Now this was competitive at that time with the rest of the globe, because in late 60s, 68, 69, people started working on prenatal diagnosis, that amniotic fluid you take through pregnant woman, you try to do prospective counseling. All the prospective counseling did not come into picture around that time, but came in mid 70s and later. And then you advise this family how to go about their genetically abnormal child. The knowledge base was not that great. Molecular biology was not in picture to be applied to this, which came in early 70s then. So I worked on that competitive area. Now coming from a small place and landing at All India Institute of Medical Sciences in Delhi, so that you can understand this is kind of a qualitative jump where you're not exposed and you want to cover that gap of coping up with those who are there parallel to you, coping up with the knowledge base which is there. So it's possible. I may be a late bloomer in that sense, but there would be many late bloomers in our country because they don't get that opportunity, that exposure. So it's important. This is another lesson. So besides you who are here, sitting out here, I would consider you privileged and very fortunate. So there are others in thousands, not in hundreds, there are who are outside, they don't have this privilege. So probably India Bioscience should also look into this issue, that scale has to be it has to be at a different scale in due course of time, if it happens. And I would just add another point over here, since I was listening to very, very informative and important, because information is power. And information is true information if you don't have. Most of the people in India do not have information. Most of the students do not have adequate information. So you are again privileged from that point of view. How this information can be, India Bioscience can play a role, because there's a gap. There's a gap that there are funding agencies, there are other agencies which are providers, but they are not in sync with institutions who are going to employ you. So there has to be a system created that they are in sync. Now, how do you create this sync with the institutions is another challenge. Job to see that how we can take it to a level where probably cure is also possible, which I don't claim at the moment, but has to be confirmed. Are there simple innovative solutions? They are at hand and you simply have to implement them. Well, you have to look for them. Probably deep, clear understanding. Because all the mentors have given you all the rules. I concur with them. I agree with them, because whatever guidelines have been mentioned by other mentors, I wanted to present it very differently through a case study of mine. And I'm coming to the conclusions of this kind, that we don't have to live in our subject domain. For me, the problem became that whenever I approach another PI that we can collaborate, they were going away rather than coming nearer. So I had to learn in 40 years all kinds of things which I have tried to show you over here. And I began as a cytogeneticist. So I had to become molecular biologist, I had to become bioinformatician, but I headed all these later because that was an advantage. I left many things undone. I feel so bad about them. I felt that they were very important to understand many things. And in the process of your movement forward, don't leave anything undone. I think right time, if you choose those questions and address them, then they will not remain undone. This is another lesson. So I regret now when I look at this list, and there will be many more, that I feel, oh, I did not do this. This could have given me a lot of dividends in the story. So what is required is, because we are always on a roller coaster, we are running. Administrator does not have, a science administrator does not have time. They're always running because they don't think, they don't stop and think. I think it's required. You don't have to be on roller coaster all the time. You have to sit with, and we say in India, you have to do manan and chintan, and probably that's also required. That's our traditional approach of meditating and then thinking. I think that keeps you cool. So that's another lesson which probably we should learn towards the end of the day, to be at peace with yourself. Because when you're at peace with yourself, you can think better. And they have been my collaborators and students who have contributed. I'm only the talking person. The rest contribution is their hard work which these students have done. Thank you very much for patience.
Okay, I shared that video because I thought that was very inspiring. I hope you found that useful. So I'm now back to sharing the slide. Is it visible? Yes, okay.
Started sharing, sir.
Okay. Has it appeared?
Not yet, sir.
Sir, I request you to please stop sharing again, reshare it, sir, then probably it will work.
Thank you.
Welcome, sir.
Any progress? Anybody able to see this so far?
No, sir.
Not yet,
No.
sir.
Subbalakshmi, can you
Yes, ma'am.
can you suggest, sir? Or sir may have to again rejoin, no? Like
Let me do that. Let me do that. Unfortunate, but let me see.
So you can completely close that PPT which we're handling. You can again reopen it and then try to
Yeah.
share it.
Okay. Okay. I'll do that.
Yes, sir, it is visible now,
It's
sir.
visible now, sir.
Okay.
Now visible.
Okay, good. So now we have come to the end, but what I want to do is to provide you some activity as a group to address a research problem, okay, or a general problem. So I will read the problem to you. Previous research suggests that 10% of the population are left-handed or sinistral is what we call it. You're going to design a study to compare two types of A-level students or simply students at 10th grade. You're aiming to see whether left-handedness is more common in some subject groups, such as art students or geographers, okay? So you want to take 10th standard students and see students who are studying arts or students who are studying geography, which type of people have more left-handedness. But generally, we know that it's around 10%, okay? So here are the tasks for you. So the task is you have to carry out this work based on observation. Of course, there is no experiment here. So you have to count the number of left-handed students, right-handed students from the two different groups, take one of them as a control. And how are you going to do this work? Whether you're going to do a survey-based work or a interview-based work. Survey is you send out a questionnaire or you sit and interview with them. So the next problem is, what are the materials you're going to use? I want you to describe the material needed for your chosen work or experiment, in this case, investigating left-handedness. Remember, if someone wanted to replicate your study, they would need to know exactly what you did, okay? Very important for reproducibility. So participants, what are your participants? The participants would be 10th grade students or let's say 11th grade or 12th grade students. Who are art students or those who are science students, you can choose them. And how are you going to investigate? So this is the activity for you. If you want, you can take a screenshot or take a picture of this. So the organizers of the workshop would like you to submit your answers or what work you've done for this activity to them. I don't know how they expect the answers to be submitted, through email or through some other means, but this is a suggested activity, hoping that you've understood some of the principles that I mentioned today and based on the talk that I gave today. But to come to the end, so is everyone clear about the activity? Have you taken a screenshot or are you able to copy this? Yes?
I have taken, sir, screenshot.
Okay, fair enough. Okay.
Yes.
So now I want to conclude by asking some key questions about the presentation that I made today, okay? So the key questions are, what is research? We learned about that, hopefully. And how to perform good research. Remember, we want to have best practices, but there is nothing called best research. Doesn't happen. So keep that in mind. There is no perfect research. So good research is what we are looking for. What is literature review, or how do we go about having a hypothesis? What are planning and management skills, or why are planning and management skills important for performing research? Number 4, how do you know you have been a good researcher at the end of a project or at the end of a career? And the example is the talk that I presented to you. So I found him to be a good researcher and a meaningful good researcher who has made meaningful contributions to the body of knowledge, okay? So I hope we were able to address some of these, may not be all, but if each one of you was able to address at least one of the questions that is mentioned here, what is meant by research, what makes a good researcher, and why research methods are important, how do we go about planning and managing research projects, and how to assess, as Dr. Bomezai was mentioning, there is self-assessment, there is peer assessment. Peer assessment can be varied, but self-assessment can be critical or simply not have any self-assessment at all. So I'm going to close here. If you have any questions, I'm prepared to take them. If you have questions later, you can email them to me or to the organizers so I can receive them. But I'm going to close here. I'm going to stop presenting so we can get back to the main window. Okay. So there we are, 6:55. Sorry if it was a little dragging or it took more time than expected. I also hope that it wasn't too boring. But if you have any questions, please ask them. Again, we're going to meet tomorrow because I have one more talk tomorrow on a related topic, which is preparing and submitting research publications or parts of a research paper. But the questions today should be focused on the.
Sir, I have one question, please.
Yes, sir.
What is the difference between research method and methodology?
Methodology is kind of, I don't know how to call it. It has become a slang. But if you are very strict, I think you should use research method as the correct term. Methodology, whenever you add that suffix ology, it's actually but methodology is what I've seen is in the Indian context, we tend to use the term methodology more common than the methods. But to be honest, I like the term method better than methodology because methodology is a study of the methods itself, or a systematic approach to the methods. I would like to put it that way. You have a systematic approach to the different methods, so that's why they may want to call it methodology. And there's nothing wrong in it, but I personally prefer research methods versus research methodology. Yeah.
Okay, thank you, sir.
There's a question here as students, this program is okay. Anybody else has any other question?
Hello, sir.
Yes, sir.
What is the difference between applied research and qualitative research, sir?
Applied research, again, qualitative research, as I mentioned, is simply you're doing research based on some observations and you're recording data. It tends to be qualitative, but you can make it applied in the sense that you can extend the study. You can make it applicable to some context. For example, if you see that you've done some qualitative research, let's say you've seen a lot of fish dying based on some observation in some highly polluted river or some pond. That's observation based, that's qualitative. So if you want to make it applied, you want to take it to the next level. If a similar thing happens over and again in different years, but the season is the same, then you have applied it to a context, which means we know that in a certain season of the year, fish die in a certain kind of water, which means there could be something happening in that body of water during that season, maybe industrial wash off during rainy season. That is what also causes fish death in a large scale. So you can make the qualitative research applied if you do the same thing over and again and extend it to different contexts or take it to a different level. You're basically making it more applied and having more people come to the same field and observe the same phenomena so that it is reproducible. It's repeatable. I hope that answers that question. It is a difficult question because the line it's a very fine line. So the question here from Varsha is, how can the researcher who is not a student can get support for his or her research? So again, that is a question for everyone. You have to seek funding to begin with from your college or universities, mostly departments. They have to fund you when they recruit. And once you have some data, of course, there are plenty of funding agencies now. It's very competitive, but there are so many programs now, as Professor Bhamesai was mentioning. We now live in a very privileged world. I say that to myself every day. And all of us who are here, I'm giving this talk, I feel very privileged to be giving this talk. And you're attending this talk. You're privileged because we have all the resources. We have internet, we have networking capabilities, we have computers. We have all kinds of privileges. We have better electricity now. In the old days, people didn't even have electricity in the colleges or universities. So there is plenty of opportunities available. So if you are a researcher, the India Bioscience website, please write down India Bioscience, do a Google search. It actually, on a regular basis, it updates the sources of funding that are available or the calls for funding that are out on a regular basis, okay? So there is a question from Sana. If you want to do research on any science related, then how should we start it out our plans, sir? So again, that you have to write it down. So a lot of things go back to the drawing room. So you have an idea in mind, you write it down, you discuss with people in your department, discuss with students, they provide input. And most important, read general science periodicals, for example, Current Science, read journals in your area, then you will get some ideas about where to go or how to start. Of course, you have to talk to people. You cannot do research in a vacuum. So you will require enormous help to perform research. Okay. Okay. Anything else? Anybody else has any other question?
Good evening, sir.
Yes, madam.
Sir, is secondary data or archival same as review article? Because say both of them have data that is taken from from other studies. Are they the same?
No, they are not. They are different because you could be using the archival material to address a question, which is a research question. But a review article is reviewing data or critically assessing data that has been presented in different research. Of course, a review article might include some data itself because they have access to some other material that the previous work did not have, but they are fundamentally different. They take different approaches. And a review article isn't written on a regular basis, but you might see archival material based research articles on a regular basis because now many people have opened up opportunities or access to research, because previously you couldn't access dead bodies that were buried. They were considered sacred, but now people allow them to be accessed. Similarly, people allowed mummies to be accessed in the pyramids. Previously, it wasn't allowed. So that is archived material, but the research is still novel. Okay, so material is archived, but your work is going to use some novel methods. So there are two different things.
Thank you, sir.
So it's let me check the time. It's I think exactly 7 o'clock now. Anybody has anything else, or the organizers want to close? It's up to you, madam.
Questions are there, they can ask, otherwise we'll end the session.
Okay.
Thank you, sir. I'm Anuradha here, and it was an excellent session and for the beginners, and also a brushing up session for the people who are already into research. And really, this particular program, which is scheduled morning 1 hour and evening 1 hour, is going so well. And it is your presence here really made a difference for the entire session. Thank you very much. And we wish we will have much more enlightenment tomorrow. And I'm sure that all the participants would have enjoyed this. Thank you, sir.
Thank you. Thank you very much. Thank you.
Thank you, sir.
Session actually for funding and all, that will be taken by Dr. Anuradha itself. So it will be at the end of the program. Yeah, I think we can conclude.
Yeah, Indu, you want to announce that thing? No, if you want to post it, you can post those competitions which we were.
Yes, ma'am. In the group, it's a PDF.
Okay.
Post it in the group.
Yes, you can do that.
Yes, sir. Ma'am, can I conclude?
Yes,
Nutana, 1 minute. Yeah.
Go ahead. Yeah.
On behalf of our college, I thank Professor Shivakumar sir for delivering an informative talk, which was very useful for all of us. Many of them have already mentioned that it was very informative and very inspiring for them as a students. And I thank all the participants who have attended this webinar. And I just want to remind you about tomorrow's schedule. Tomorrow we have 2 sessions in the evening. 5 to 6 will be taken by Sushil Kumar sir. And 6 to 7, we have again the session with Professor Shivakumar sir. So please do attend and gain the knowledge. Thank you.
Okay, thank you, ma'am. Okay.
Yeah, the feedback form is posted in the chat box. Please give your.
Okay, thank you.
Thank you, ma'am.
Thank you, one and all.